Hello, everyone, and welcome to this VCCC Monday Lunch Live series. I hope everyone is able to hear me. My name is Fanny Francini, and I'm a research fellow at the University of Melbourne, and today I will be sharing this session. Firstly, I would like to acknowledge the traditional custodians of the lands on which myself, Dr. Koen Degeling, and the audience is located. I would also like to pay my respects to the elders, both past and present, and extend that respect to other Indigenous Australians who are present online today. Before we kick off today's program, there are a few housekeeping notes that I would like to share. So the VCCC has some interesting webinars that are coming up this month and in May, and you can register via the VCCC website to attend these. Please note that today's event is being recorded, and this will be available on the VCCC Open Learning page, where you can also view previous webinars. And of course, you can follow the VCCC on social media on Facebook, Twitter, and LinkedIn. You've got the @mentions here. And don't forget to tag the VCCC in your posts. And if you have any question for today's presenter, you can do this via the Zoom Q&A function as shown on the slides. And we encourage you to submit your question throughout the presentations, and we will aim to answer your questions at the end of the session. So our speaker today is Dr. Koen Degeling, and it is my pleasure to introduce Koen, who is an outstanding researcher, but also a wonderful colleague. So Koen is a research fellow in Cancer Health Services Research within the Center for Cancer Research and Center for Health Policy of the University of Melbourne. His research focuses on the use of advanced simulation methods to model personalized oncology care pathways, with a particular interest in the use of real-world data from clinical registries. Koen is involved in several research projects across the VCCC, and he received very recently the 2020 Rising Star Award from the Melbourne School of Population and Global Health. And this was in recognition of the extent and the impact of his recent work, which includes the use of simulation models to estimate the impact of the COVID-19 pandemic on long-term outcomes for cancer patients. And so today, Koen will present on the concept of simulation models and demonstrate their role in health technology assessment and beyond. So we're very much looking forward to hearing Koen's presentation.
Thank you, Fanny, for that kind introduction, and thank you to the VCCC for the invitation to present to you today. It's a great honor to present about the use of simulation models to optimize the development and use of oncology innovations. So as Fanny said, I'm part of the Cancer Health Services Research Unit of the University of Melbourne. And that's the unit that's being chaired by Professor Martin Eisenman. And as you can see on this picture, we're located in the VCCC building. And even though the Cancer Health Services Research Group is relatively young, I think next week we celebrate the 3rd birthday, we work on a quite substantial research agenda across 3 main research priorities, being the health economics of cancer genomics, real-world data from clinical registries to evaluate health services, as well as a data-driven approach towards improving access to care and informed cancer services. And the reason why I highlight this is because today, during my presentation, I hope to take you on a journey across these 3 priorities and show how we use simulation modeling across them. So I'll start with a quick introduction to the concept of simulation models before moving on to the more traditional application to evaluate healthcare interventions. And then we will go into some newly waters, being the use of simulation models to optimize development, as well as explore how we might use them to inform oncology care. So let's start with the concept of simulation models. So what they are. Well, in general, I think all models are can be summarized as simplified representations of reality. And what I mean with that is that typically we develop a simulation model or a model in general to answer a specific question. And to do so, we use evidence that's relevant to the question we want to answer to define the model input parameters, as well as to define the model structure. And in the work that we do, this structure typically represents a clinical or disease pathway. And then what we do is that we simulate hypothetical individuals that run through this structure and experience events. And by capturing these events and their consequences they may have, we can capture outcomes, which typically include life years, quality adjusted life years or QALYs, and for example, toxicities. And if it's a health economic study we're performing, we also capture costs. And I understand that this is quite an abstract example, but throughout this presentation, there will be more specific ones. So a question that I often get is, what is a model and what is a simulation model? So what's the difference between the two? Well, when I speak about models in general, I'm referring to two specific types. So first of all, we have the cohort level models or mathematical models that actually calculate the outcome for an average individual or an average patient. And these are the methods that traditionally have been used. But because they take this sort of average approach, it's quite hard to capture complex and dynamic processes, such as personalized medicine processes. So that's where simulation models come in very handy. So simulation models are individual level models, which means that rather than simulating or calculating outcomes from one average patient, we simulate hypothetical individuals, each of which have their own characteristics, history, and outcomes. So it allows us to represent heterogeneity as well as stochasticity. And because they are more flexible, and because we can allow this heterogeneity, simulation methods allow us to more naturally represent the complex dynamics of clinical pathway. And now that computing power is ever increasing, it's very feasible for us to use these methods. So that's in a nutshell what they are, but let's have a look at what they actually are useful for. So one example that some of you may know, but some of you may also be not that familiar with, is the use of simulation models in hospital logistics. So we can use them to evaluate, for example, how an X-ray MRI scanner would impact the patient flow in a hospital, or how alternative resourcing of an emergency department would impact waiting times and research utilization. This hospital logistics is not something that we mainly focus on within the Cancer Health Services unit. We're more looking into the health technology assessment, where simulation models come into are very useful as well. So to evaluate healthcare technologies, simulation models are very useful to extrapolate evidence, as well as to combine evidence from different sources. And in today's presentation, I hope to share with you some examples of how we can even go beyond that and use simulation models, for example, to obtain insights about the required performance of the new biomarker test, or to inform optimal treatment sequencing for subgroups of patients. And by no means this list is exhaustive. There are many other applications, and I think everyone has become familiar with the use of simulation methods to model infectious diseases, like the models that our colleagues at the University of Melbourne have developed to inform the response to the COVID pandemic. And we ourselves, with a group of VCCC experts, have also used models to estimate the impact of the COVID pandemic for long-term outcomes for cancer patients. So there's many different applications where these models may come in useful. All right, so that's simulation models in a nutshell. Let's have a look at how they've been traditionally being used to evaluate healthcare interventions. And for this, I'll discuss an example in favorable or low and favorable risk localized prostate cancer. And in this setting, there is basically two choices between decisive or local upfront treatment or an initial active monitoring or surveillance strategy. And the decisive or radical treatments, such as radical prostatectomy and radiotherapy, they take action against the cancer. But a potential downside of them is that they are associated with common complications, such as urinary, sexual, or bowel complications. Active surveillance or active monitoring, on the other hand, is the concept of carefully monitoring the disease and thereby delaying or maybe even avoiding radical treatment. However, because you delay taking action against the cancer, there may be an increased risk of disease progression. So to answer the question, what is the best treatment option in this setting? The PROTECT trial was performed in the United Kingdom, and today this is the only study that randomized about over 1,600 patients to either active surveillance or active monitoring, radical prostatectomy, or radiotherapy. And what this trial showed us was that if we, for example, look at the proportion of patients receiving treatment over time, with in green radical prostatectomy, in blue radiotherapy, and in the black dotted line active monitoring or surveillance, you see that on the monitoring arm, there's a continuing increase of patients who will receive treatment over time. And on the other hand, if we look at the progression-free survival, you see that for the active surveillance arm, there was an increased risk of progression. However, this did not translate into a difference in overall survival. So this poses a very interesting question, because the difference between the proportion of patients being treated suggests that those on active surveillance may have fewer complications, but the progression-free survival curve also shows that they may be, or that they are at an increased risk of progression. So the trial group also performed a cost-effectiveness analysis based on the trial data, and they found that active surveillance or active monitoring was less costly over the 10-year time horizon of the trial, and that the outcomes in terms of quality-adjusted life years were similar. But the big question is, what happens after the 10 years of the trial? Because as we saw in the figures, there was a clear increase in the patients on the active monitoring arm who received treatment. So they may be exposed to the complications of treatment at a later point. Similarly, or at the same time, the differences in the risk of progression seem to increase over time. So the important question is, what happens after the trial horizon? And it's also a question that was asked by the trial authors. So what we did as part of the Prostate Cancer Prognosis and Treatment Project, or PRECEPT, which is being led by Neil Cochran, who's the VCCC Research and Education Lead in this area, we developed a simulation model that combined all the best evidence we could find on the prostate cancer process. So as you can see in this model schematic on high level, the model includes everything from the diagnosis, but the active surveillance, the radical treatment, treatment monitoring, management of progressive disease, and development and treatment of metastatic disease. And we used data from the PROTECT trial to extrapolate the probability that patients would receive treatment over time. Based on that, we also from the PROTECT trial took the incidence of complications and the impact they had on the quality of life of the patients. We extrapolated the progression-free survival curve. And as you may know, there's different methods to do so. So we had to work closely with the team of clinicians involved to select the extrapolation that was considered most appropriate. And then we modeled the risk of developing metastatic disease after progression. And that's the point where the PROTECT study sort of got too limited in the amount of follow-up, because there were relatively few patients who developed metastases, and they were followed up for a short period of time, and their detailed results were not presented. We used evidence from different randomized trials in the metastatic setting, so the CHARTED and GTUC trials, to extrapolate overall survival for different treatments and different subgroups of patients separately. Then we modeled background mortality based on the Australian live tables. And finally, because we were also interested in the cost side of things, we implemented costs in the models based on the MBS, the PBS, and the National Hospital cost data collection from Australia. So what allows this us to do? Well, if we look at the short-term outcomes, which are not presented on this slide, but when we looked at the model for a time horizon of 6 to 10 years, we indeed found the same as what the trial found, fortunately, that active surveillance was more cost effective on the short term. However, we could use this model to look at the lifetime of the patients. And what we saw that on a lifetime horizon, active surveillance was more costly than radical upfront treatment. And the reasons for that were threefold. So first of all, over the lifetime, a substantial proportion of patients on active surveillance ended up having treatment anyway, and therefore exposed to the associated risk of complications and also the treatment costs. At the same time, they were subject to a higher risk of progression due to the delayed treatment. And as you can see on the right side, treatment of progression as well as metastatic disease contributed to a substantial increase in costs. In the end, turning out that active surveillance, therefore, was more costly. If we look at the health outcomes, or the quality adjusted life years, we again found that there wasn't a very big difference, or basically no difference, between the strategies. And that suggests that the tools that are used to measure quality of life in these patients are not sensitive to the treatment complications. So I hope this is a clear but also traditional illustration of how simulation models come in hand useful to combine evidence from different sources and provide insights beyond the time horizon of a clinical trial. So now let's look at a more innovative application of simulation models, where we extended the previous model to inform the development of a new biomarker in this setting. So in the previous slides, we basically found that active surveillance may not be cost effective for the overall low and favorable risk localized prostate cancer population. And that's from a health economic perspective, of course. But that doesn't mean that there's very likely a subgroup of patients for whom active surveillance is the most cost-effective option. And therefore, there's a substantial interest in the use of biomarkers to improve risk stratification, to not only reduce undertreatment, but also reduce overtreatment. Our colleagues and as we recently performed a systematic review of the cost effectiveness or health economic evidence in this setting, and we found that even though there was health economic evidence for 17 biomarkers, 7 of which aim to inform this first-line treatment decision, the cost effectiveness estimates were very uncertain. And the main cause of that was that there was limited evidence on the clinical utility of these biomarkers. So they were mostly based on validation studies only. And it's relevant, or the lack of certainty in these estimates is very relevant because many countries, including Australia, require evidence on the cost effectiveness of such new tests before they are reimbursed by public healthcare system. And because there is no solid evidence on this, you see that the biomarkers are mainly used routinely in the United States and not elsewhere. So given that in the PRESET project, the aim is to develop biomarkers in this setting as well, we wondered what conceptually the potential is for these biomarkers to improve health outcomes in a cost-effective way, and what are priorities in the development of these biomarkers. So what we did is we took the previous model and we extended it to include something that we call here a treatment decision module, which includes the simulation of biomarker test results and their use by patients and clinicians. So this is step one in this sort of module, and I won't go into a lot of detail here, but conceptually what it does is that based on what the initial treatment recommendation would have been, it simulates test results and how then the clinician used this test result to inform their recommendation about treatment to the patient. And subsequently, the patient uses this information about the recommendation by the clinician and the biomarker to make the final treatment decision. And this was modeled in quite a granular level. The reason for that is that it allowed us to evaluate different scenarios. So we could simulate different biomarker profiles based on, let's say, sensitivity and specificity, but then also how patients and clinicians would use them. So how likely was a biomarker to result in a de-escalated or an escalated treatment decision. So we use this very flexible model to evaluate a lot of different biomarker scenarios. So let's have a look at some of the results. But first, we should take some time to walk through this figure because there's a lot of information in it. So what we are looking at in general is the health economic impact of a strategy that uses a biomarker test to inform treatment compared to a strategy where no biomarker is used. And then the outcome here is the net monetary benefit, or NMB. And it's a health economic measure that combines both health outcomes and costs. And basically anything that's positive is considered cost effective. And to get to this summary measure, we need to assume a willingness to pay. So how much money society is willing to pay for 1 year in perfect health. And here we used 20,000 Australian dollars per quality adjusted life year. And that's quite a conservative estimate. So that means that everything in green here is something that's really cost effective. Okay, then about the figure itself. So on the vertical axis, so this figure looks at the diagnostic sort of performance of the biomarker test. And on the vertical axis, we see the test sensitivity. And on the horizontal axis, we see the specificity. And on the left, you see the outcomes for if the biomarker would predict whether there may be progression on active surveillance. And on the right, you see the results for a conceptual biomarker that predicts whether the patient will develop metastatic disease when on active surveillance. And then for this scenario, what we assumed furthermore is that the compliance was 100%. And that means that if the test would recommend active surveillance, then patients and clinicians will actually select active surveillance. So there was perfect adherence to the test result. And the test was relatively cheap at 1,000 Australian dollars. So this is a sort of the best case scenario, let's say. All right, so what do these results show us? Well, overall, there's a lot of green. So that means that there's there's good health economic potential for biomarkers to improve health outcomes in a cost-effective way for localized prostate cancer. And then if we compare the left to the right, we can see that there is more potential for these biomarkers if they predict whether the patient will develop progression, yes or no. And if we think about the results of the previous study, that makes sense, because what we saw there is that the treatment of progression and metastatic disease is very expensive. And as well, if patients progress, then their quality of life deteriorates as well. So what we want to do is to prevent progression from happening. And if you only want to prevent metastases from happening, then there will be a proportion of patients that will progress, but not to the metastatic phase. So from a health economic perspective, these results suggest that we should prevent undertreatment as well as overtreatment, but also that undertreatment is really important to prevent. Additionally, if we compare sensitivity to specificity, we can see that the test sensitivity has a much more profound impact on the cost effectiveness. So the test sensitivity can really change the outcomes from not cost effective to cost effective, where the test specificity not really does so. And this again is in line with previous findings where we saw that preventing undertreatment is very important. But this is really a technical view on sort of the profile of the biomarker, so the technical requirements. But we can also have a look on how the test is being used in practice and how that then influences the outcomes. So this is a similar figure, but here we assume a test sensitivity of 90% for an area under the curve of 0.8 and the same price. But now on the vertical axis, we have a look at the compliance to an escalation recommendation. So this is when the test recommends that there should be a decisive or radical treatment. And on the horizontal axis, the probabilities that patients and clinicians follow a treatment de-escalation recommendation. So if the biomarker suggests that active surveillance is a safe option. And well, what this clearly shows is, well, first of all, again, that there is a lot of potential for tests to inform whether there may be progression or not, but also that compliance with escalation is more important from a cost effectiveness, so health economic point of view, than compliance to the de-escalation. And this is in line with what we saw with regard to the sensitivity and specificity, because this suggests that preventing undertreatment is very important, as well as preventing overtreatment. And these are, well, at least I find quite interesting findings on that, because in literature, the tendency has been about reducing overtreatment and not as much on reducing undertreatment. But these results suggest there is substantial opportunity for biomarkers to add substantial value by preventing undertreatment as well. But again, this is only one sort of part of the equation. So this solely looks at the use of the test, but we can also look into more the technical requirements and the use of the test at the same time. So this is a slide with a lot of information, and it's not something you have to fully comprehend, but I want to share some of the main findings here. So this figure shows the impact of the test costs in the columns, then whether the test predicts progression or metastatic disease in the rows, and then the test performance, so sensitivity and corresponding specificity based on this AUC. And on the horizontal axis, combine the compliance with escalation and de-escalation. And first of all, if we consider that the tests that are commercially available in the US cost about $5,000 US, it's very unlikely that this will be cost effective. But you'll still see a lot of green in this figure. And we've used a very conservative willingness to pay or cost effectiveness threshold. So there's fortunately a lot of potential for biomarkers to add value in this space. But then if we look to sort of the trade-off between the sensitivity of the test and the compliance, it's very interesting to see that they're both very important. And that test sensitivity is important, I think, well, everyone would have expected, but that the impact of how the test is being used in practice is as equally as important, maybe, well, at least I find very, very interesting, which suggests that we can invest a lot in the development or improving the diagnostic performance of a test. But if we don't as well invest in making sure that the test is being used as intended, it will very unlikely be successful in achieving its aim. And of course, this is based on the simulation model, so it's only insights that we obtain. But I find it's pretty interesting insights and very useful in the development and translation of such a biomarker. So that concludes this example on the use of simulation models to inform the development of oncology innovations, which I hope you found interesting. But let's take this all one step further and see whether and how we can use simulation models to actually inform oncology care. So for this, we will make a switch to the setting of metastatic colorectal cancer. And on this slide, you see a so-called Sankey diagram that visualizes the treatment sequences for a subset of patients from the treatment of recurrent and advanced colorectal cancer, or TRAC registry. And even though people may say that the treatment landscape for metastatic colorectal cancer is less complex compared to other metastatic cancers, it still looks quite complex to me. And that brought me to the question, can we use all the information in this real-world data together with simulation models to inform what is the best first-line treatment in this setting? And I'll get to what I mean with best here. But because this was a very exploratory study, we wanted to start relatively simple. So we wanted to zoom in on one specific question. And looking into the data, we saw that about 72% of patients either start treatment with doublet chemotherapy or doublet chemotherapy with bevacizumab. So our research question was whether we could inform this decision based on the real-world data using simulation methods. And if we think of this question, then on a population level, so on average, there is still consensus that doublet chemotherapy with bevacizumab may be the best option. But may also be a subgroup of patients for whom that may not be the case. And it also depends what you mean with best. Is that best in terms of overall survival, progression-free survival, or is the another outcome that's important in making this judgment? So we wanted to look into simulation models to see whether we could provide some information here. So this is the conceptual structure of the simulation model that we developed. And as you can see, there was a first-line treatment here. And then subsequently, there was the option for further treatment or best supportive care. And even though this structure may seem more example than the prostate cancer model I just presented, internally it's more complex. And the reason for that is that in this simulation model, we in very much detail accounted for patient variation in terms of characteristics, treatment history, as well as treatment outcomes. And so what we could get from this is not only overall survival, but also first-line progression-free survival, the likelihood that the patient would go on to further treatment, and then overall survival conditional on that, so for second-line treatment and beyond, as well as, of course, overall survival after all. So in order to achieve that, we modeled progression-free survival, of course, based on what treatment was provided, the intent of treatment, and a range of patient characteristics, such as the stage at the initial diagnosis. And then based on that, we modeled the probability of further treatment. But in this case, we also included the outcomes of first-line treatment. So the progression-free survival that was achieved on first-line treatment, and then again, the treatment and patient characteristics. Again, conditional on what happened before and the treatment being provided in the future, we then modeled overall survival after first-line progression. And the value that we add here is that we take outcomes of previous treatment lines into account when predicting what may happen in the future or modeling what happens in the future. And although that provides us with enhanced information of all these different outcomes, it's also much more complex to model. And it takes a lot of time to consider how you would, for example, best model overall survival after first line based on first line progression. And I think you can best compare this to a relay race, because if one of those parts of your model break down, the overall result will be poor. And it's the same with the relay race. Like all runners except one may be exceptionally good. But if there is one runner who doesn't yet make it, then the overall result will not be as intended. And that's for the simulation model. The validation is very important, but also more complex than what we're used to. And this is just one example that I would like to show of the validation that we've done. So what it shows here is that for different risk groups of patients, and with regard to first-line progression-free survival, for example, it predicts in dark gray. It shows what the simulation predicted the risk of progression was at 1 year. And then in light gray, you see what for those patients in the data, the Kaplan-Meier estimate was. And you can do that for different sizes of subgroups, as well as for the different outcomes. And this is an example of model calibration, but we also looked into model discrimination and other things. But overall, we were quite happy with the performance of the simulation model. I do want to emphasize that even though this was bias-corrected internal validation, it's still internal validation. So before we ever want to consider using things like this in practice, we will have to externally validate the models, which means that we have to validate its use in a group of patients that were not used to train the simulation model. But let's see what this allows us to do conceptually. So it allows us to run a so-called personalized simulation. So we can take a certain patient profile, for example, based on age, stage of diagnosis, and other characteristics, and run the model for both strategies. So doublet chemotherapy, or the experimental or alternative strategy, doublet chemotherapy with bevacizumab. And then for both strategies, we'll get this set of outcomes. So first-line progression, the probability of further treatment, and overall survival. And that can potentially then inform a treatment decision. But because we have information on these multiple outcomes, another question is, what then determines what the best treatment is? So relative weight of these different outcomes are very likely unequal in the importance with which they inform decisions, but how important they are may also differ between patients and clinicians. So although this doesn't allow us very easily to optimize the best treatment, we can do some interesting exploratory analysis with them. So one thing we did is we performed an example where we aim to maximize progression-free survival. And that's the results of which you see here. So what we did here is we ran the whole patient population from the track registry that had either doublet chemotherapy with or without bevacizumab. We performed a personalized simulation for each of them. And then we compared in black the treatment they received in real life to what the simulation model predicted to be the treatment that maximized progression-free survival. And for those who switched treatment, so for whom this simulation predicted that another treatment may be better, which was 25% in this case, you see a clear difference in the outcomes. But if we look at the total population, the difference was much smaller. And this is by no means to suggest that those patients didn't receive the right treatment. This is just an illustration of that this conceptually could be used to perform some optimization exercises. And this only looked at progression-free survival, whereas we all know that in real life, there's much more to a decision than progression-free survival. But it's just to illustrate what potentially we may be able to do after such a model is developed and externally validated. So I think my general takeaway from this all is that at least I find it very exciting. In oncology, most prediction models have focused on predicting overall survival, such as nomograms and now increasingly the machine learning algorithms. But with these simulation models, we're able to combine those together for different outcomes in one framework and combine evidence from different sources. So not one big data set on which we have to train, let's say, a machine learning algorithm. And I think another great advantage of these simulation models is, like the ones we used, we used survival models to populate the simulation, is that those survival models are not black boxes like most machine learning algorithms. So they're much more like standard regression models, where we can understand what the impact of all covariates are on the outcomes and whether that actually makes clinical sense. So it's a very flexible framework to combine all the knowledge we have in data, in literature, or in clinical expertise. But there's still a long way to go. And I think some of the key questions that we have to look into beyond doing more of these explorations and demonstrations and external validation, there's a few very conceptual but very important questions we have to consider. So first of all, what additional outcomes than those that I presented should those models present? For example, quality of life. And then what level of detail should these simulation models provide? Should it go into the level of detail of whether metastases were resected or not? And if we think about systemic treatments, how many lines of treatment do these models have to account for? And in what level of detail? Is it sufficient if we look into the question, or if we look into sort of groups like doublet chemotherapy? Or do we have to look into whether those are irinotecan or oxaliplatin based? Or do we even need to go one step further and look into whether the chemo backbone is fluorouracil or capecitabine? And then if we know what outcomes we want and the level of detail we want to achieve, do we actually have sufficient data to achieve that required level of detail? And then let's say we have, then we have a lot of information coming out of these simulation models, but how then do we combine that into, in a way to actually inform decision-making in a meaningful manner? So for example, could we then map those simulation outcomes to the value frameworks developed by ESCO and ESMO? And very important, how good should these simulation models be? So what's their performance for them to be used in clinical practice? But also, how are we going to measure that? What statistical methods are we going to use? Or do we have to perform, let's say, clinical studies on these simulation models as well as decision tools? And then finally, even though these models are not black boxes, they become very complex. And how complex can they become for us to still have sufficient confidence in that they do what we think they do? So a lot of exciting opportunities, but also very important questions that we all have to consider. And I think these questions are, well, maybe are even more applicable to the clinical and patient side than they are really for us modelers to answer. Because in the end, it's the clinicians and the patients who we aim to support in their decisions. So they should be comfortable and confident in using such approaches. So it's a far way to go, but a very exciting field, at least to me. And well, with that, I've completed my journey on using simulation models to optimize the development and use of oncology innovations. And lastly, I would like to thank my collaborators on these projects that I've presented about, which for the metastatic colorectal cancer work is the personalized oncology division at WeHive, Peter Gibbs and colleagues. With regard to the prostate cancer work, it's the PRECEPT research team, including the affiliated researchers, for example, at Peter Mac, and especially also the VCCC consumer representatives who are actively engaged with the PRECEPT program and provided very important input on conceptualizing the simulation model that we used. And then finally, my colleagues within the Cancer Health Services Research Unit that contributed to this work, and especially Professor Martin Eisenman, who, in my opinion, built a very unique group with substantial and meaningful output in such a short period of time. And I'm very grateful for the environment that he's created for me and my colleagues to do what we do best, and that's our research. So thank you for your attention, and I'm happy to take any questions.
Thank you, Koen, for this great presentation, which showcased the use of simulation models to optimize the development and use of oncology innovations. I really enjoyed the talk and the animation and illustrations that you have used throughout to bring perspective to your work. So I will start with questions that were asked in the chat and Q&A section before follow with verbal questions if you would prefer to ask them live. So we have 3 questions in the Q&A. So I will start with questions from Professor Grant MacArthur. So great talk, Koen. How would you see modeling working in a learning health system in tandem with real-world data?
Thanks, Brian. So I think traditionally data and models have been seen as 2 separate things. So sort of there was a once-off transition of data to inform the model, and then there was a separate entity. But I think with, well, as quickly as the registries are developing, I think that we can no longer see them separately. And the simulation models are so dependent on data that I can envision that, well, in let's say 5 years' time, every registry also has a simulation model that sort of lives on top of it, and that routinely, let's say every night, the parameters get updated based on new data that may be entered into the registry. So I think that this sort of, well, once-off transaction will turn more into a dynamic and continuous process. At least is what I would hope to achieve.
That would be amazing. Another question from Grant. So how can the patient and consumer voice can be integrated into the modeling studies?
Yeah, that's a very difficult question and something that we actively try. And I think we find good ways because, well, originally, I think the modeling has been always highly technical. But I think that the consumer input is very important in defining the scope of the model. So, for example, in the Precept study, we worked together with Sophie, Les, and Keith from the 3CCC Cancer Consumer Advisory Committee on what the model should present in terms of outcomes, but also what was important to be included in the model. So we discussed which complications, treatment-related complications we had to account for. And then for every complication, there is like different measures that were used by the published studies. So we went through those measures together with them to select the one that they found most relevant. And that's very important input, let's say, on the level of detail and on the outcomes. That's very hard for me as a modeler to answer, but where consumer input is really critical.
We have another question from Professor Mark Jenkins. So for the prostate cancer modeling, where you speak about the monitoring versus surgery versus radiotherapy. So what dictated which patients initially allocated to monitoring, went on to surgery or radiotherapy, and how important is that for the modeling results?
Thank you, Mark. So for the first study I presented where I compared the 3 strategies, so active surveillance, radical prostatectomy, radiotherapy, it was 3 scenarios. And in each scenario, 100% of the patients would go to active surveillance, radical prostatectomy, radiotherapy. For the biomarker, we based the initial allocation based on real-world data for Australia. So for the cancer registry of Victoria has published the proportion of patients for low-risk disease and favorable intermediate-risk disease who in current practice opt for active surveillance or one of the decisive treatments. And that's the F we use to inform those, well, let's call it prior distributions. And then conditionally on that, we simulated the use of the biomarker and how that may influence those proportions. Great.
Another question from Jianrong in the chat. So what Jianrong understands from the simulation model is that each model really depends on the context of the regions, like the cost of intervention, cancer epidemiological patterns, and the option of interventions in the region. So therefore, how to consider the generalizability of the models?
Yes, thanks for that question. No, that's absolutely right. So we found that there's been a lot of research in the generalizability of economic outcome estimates, and definitely everything that relates to the costing and the resource utilization within healthcare systems is very different between countries. So for work, we used a Protect study, which is from the UK, and even though there may be similarities between the UK system and the Australian system, they're also quite different with regard to a lot of aspects. So what's being often done is that the very hard sort of efficacy data is transferred between countries, but then all the downstream impacts such as like the value for the quality of life estimates, the resource utilization, the costs, that's all specific to the country. So that's what we've done here as well.
Great. We have another question from Nusad Fajri. So how do you see the use of these simulation models with other surrogates outcomes, such as overall response rates, event-free survival, etc.? Would it be similar to OS and PFS?
Thank you, Musa. Well, that's sort of unexplored terrain, and that's something I'm very interested in, because when you think about such detailed outcomes, you also have to think about the sequence in which you estimate them if you want them to influence each other. And that's a question that I hope to look into with the use of real-world data that we have available. So to see how different approaches impact outcomes and the accuracy of the estimates. But currently there is no golden standard or even like guidance on how to do it. So that's an opportunity for further research that I'm keen to explore.
And as you mentioned, you need to have the data available as well.
Yes, definitely.
Which is
yeah.
really crucial. I did have a question that I still want to ask. So you have shared three main categories where simulation models are used in your research. And the first one you presented is the more traditional approach that's used, in research. But I just wondered, for the other applications that you have showed today, are they somewhat mainstream? Is there a large body of work that's being done, or are you really leading the research? And how do you see the research developing in the future?
Yeah, so I think sort of my second example, which is like how we can use models to inform technology development, is something that's quite well established as well. And it's often referred to as early health technology assessment. Where typically the aim is to establish the potential or headroom or room for improvement for new technologies to improve outcomes, and then how expensive that may be. So that's quite well established. The thing that we add there is that typically those calculations are quite high level average, so they use limited data. And in the example that I showed, we really just developed a very detailed model to get the flexibility to evaluate all those different scenarios and see how, for example, use by clinicians and patient influences outcomes. Then the last example, so to actually make the step to improve oncology care, that's very innovative. And the paper I presented about, we published last year. And as far as I know, it's the first who really went into that direction for oncology. There have been similar models in other fields of research, like I believe there are some in rheumatoid arthritis, as well as some HIV, I believe. But applications are very limited. So that's definitely a developing body of work.
Amazing. So lots of opportunities.
Definitely.
All right. If there are any other questions, let me know whether you would like to ask them live.
Yeah, I think there's Martin had a question.
Oh, I don't see. Sorry.
It's in the Q&A.
Yeah. So Martin Degeling has a question as well. So we are now mainly developing and applying these simulation models to improve care. So, but what do you think is required to actually change clinical practice? Do you have examples where these models have informed and changed clinical decisions? And what should be done to improve this?
Yeah, that's the key thing, right? Because we can develop all these, well, what we think amazing simulation models, but then if they're not actually contributing to improved care, then yeah, that's a very well missed opportunity. And that's also the challenging part. So I think the questions I highlighted at the end to get the dialogue started about that. So what do we really expect from these models in order to be used to inform clinical care? I think that's a key next step. And I think a lot of oncologists have used, for example, nomograms to well, at least communicate risks to patients. So the concept of using such model-based estimates is not new, but the simulation models become very much more complex and give more information. So I think we should really start to have a conversation about that. And it may even require us to do some pilot studies where we, in a safe environment, but see how patients and clinicians value such simulation models and whether they actually have the potential to change decisions. Because even though the information may be helpful, if they don't change actual decisions being made, then they will not improve outcomes.
Yeah, that's true. And speaking of outcomes, so another question. So thanks for a great talk. Can you expand on the external validation of the model for treatment of metastatic colorectal cancer? And for instance, what patient size do you need to do this type of validation?
Yeah, well, thank you. So what we hope to do is at some point, because I think we use data up to 2017 from the TRACK registry to inform the simulation model. In about a year's time, I guess that there's a substantial cohort of subsequent patients that we could use to do external validation. And that's then also the most recent patient cohort. So I think that's a great opportunity. In terms of what patient size we need, I think the number of patients we used to develop this simulation model was about 800 something. So it's not, well, it's very huge numbers, but it's not like tens of thousands. But I think a couple of hundred patients are definitely required because we're looking into so many different patient subgroups. If you then split them up in those different risk categories, the numbers per subgroup become small quite quickly. So yeah, a couple of hundred patients would definitely be required to get at least some confidence in the validation estimates.
Great. Apologies to Martin and the anonymous attendee who asked the last question. I missed the last two questions you just asked, but I see no other further questions. So I think we can conclude the seminar. So a slide should appear. So that brings us to the end of today's Monday lunch live stream webinar. So thank you to Koen again and for the and to the audience for tuning in. And please, you can tune in next week for the next live stream, which will be titled Investigator-Initiated Trials Innovations. So it should be quite good as well. Thank you to everyone.